Hello, this is Emilia, and I'm going to talk about the regulation of pollen and nectar foraging in honeybees and the changes in gustatory perception, learning, and memory in bees arriving or departing from food sources. Well, in honeybees, the collection of food sources, mainly protein and carbohydrates, is achieved by individuals specializing in pollen or nectar foraging. Foraging task specialization is linked to differences in bee sensitivity to flowers' rewards. In behavioral bioassays, using the proboscis extension reflex or pair, an innate response elicited when sucrose solution contacts the antenna, researchers measured the gustatory sensitivity of pollen and nectar foragers by the successive offering of increasing concentrations of sucrose solution. The results showed that pollen foragers are more sensitive to sucrose than nectar foragers. In other studies, where the pair was also used as response variable, researchers conditioned bees to the association between a stimuli, that could be a color, an odor, a texture, and a sugar reward. As pollen foragers are less demanding regarding sugar rewards, they also learned better during associative learning. So far, differences in gustatory perception have been observed between nectar and pollen foragers returning to the hive, but have not been studied yet in bees at the beginning of their foraging visit, when they are highly motivated to forage. Our objective is to study the changes in gustatory sensitivity between pollen and nectar foragers when arriving and departing from food sources. Our hypothesis is that pollen foragers modify their gustatory sensitivity according to the moment of the foraging visit. For this, we captured foragers when they arrived and when they departed from feeders that contained sucrose solution or pollen. Then we measured the gustatory sensitivity of foragers by the increasing and successive presentation of sucrose solution. Moreover, we olfactory conditioned foragers to study differences in acquisition and retention of the association odor sucrose and the association odor sucrose plus pollen. In terms of gustatory sensitivity between pollen and nectar foragers that arrived and departed from the feeders, in the figure, the x-axis shows the different concentrations of sucrose and the y-axis the proportion of pair. Our results show that nectar foragers were more sensitive to sucrose than pollen foragers when they arrived to the feeder. As we knew, pollen foragers were more sensitive to sucrose than nectar foragers when they departed from the feeder. Also, pollen foragers that departed from the feeder were more sensitive than pollen foragers that arrived to the feeder. In terms of learning and memory of pollen foragers that arrive and pollen foragers that departed from the feeders, the figure, the x-axis shows the learning events or trials, and the y-axis shows the proportion of pair. Our results show that pollen foragers that arrived to the feeder learned and retained olfactory memories better with a dual reward, sucrose plus pollen, and that pollen foragers that departed from the feeder learned and retained olfactory memories in a similar way with sucrose or with sucrose plus pollen. Our conclusions are that differences in stimuli sensitivity are determinant for task allocation between foragers. Low sucrose responsiveness at the beginning of the foraging visit would prevent pollen foragers from being attracted to nectar sources. And pollen foragers that arrived learned saucer's cue better when are reinforced with pollen. Hence, they are more sensitive to pollen at the beginning of the visit, while at the end of the visit, pollen may difficult or even perturb learning. Thank you for your attention.